back. We're here. Today is likable, even though it's raining a storm out there. Whoa, so bad. Anyway, this is Likable Science, and you may wonder if you lost your true vision here, because actually Ethan Allen is the host of Likable Science, and I am not the host of Likable Science. But I'm sitting in the host's chair this time for Likable Science because I can't answer any questions about this, and he can.
But Jay, this is all sort of you're doing. You put us on to this article and discussed it.
I put it on to you, but that's as far as it went. So Ethan is the host, and he is a likable scientist. That's why we call this Likable Science, because of Ethan. He's a scientist with PREL, the Pacific
Resources for Education and Learning.
Okay? And the name of our show has a lot to do with RNA. And if you didn't know from your high school biology or wherever you might have learned it, RNA is ribonucleic acid.
Very good.
R for ribo, N for nucleic, and A for acid.
Exactly.
As opposed to DNA, which is disoxyribonucleic acid.
Close.
Rather D for the D in DNA,
Right.
and then N for the nucleic and A for the acid. It's all about acid. Okay, but before we get, the title of our show is, listen to this, is RNA the Way to a Better Day? And that's a serious question, and that's our show. And if that leaves you in a cliffhanger, good, because I have some housekeeping. Number one is if you want to send us a question or a comment by Twitter, you can do that on ThinkTech HI. That's our Twitter handle. There it is on the screen. Secondly, take a look at our website these days. It's in the process of being improved and revised, and there's much more on it, and there will still be more on it, but it is different than it was. So take a look at thinktechhawaii.com, okay? And let's see, that's all I have. There's one other thing in the way of, okay, this just in, Ethan is going to be doing a science commentary program, science briefs, if you will. He's going to be doing them probably on some day other than Friday. We're not sure yet.
Okay.
But let me say that if it's on Thursday, it's going to be called Science Thursday. We like to get the jump on the other guys. You don't look like Ira Flater. Okay, now to serious. Okay. We have been dealing with GMOs, which is, what is GMOs? Genetically modified organisms for a long time. They must have been invented way back when, the 30s probably, where we can modify something and change, mutate it scientifically at the molecular level, the cellular level, I guess, by high tech. And this is really fabulous, and it has led to the creation of a whole new agricultural industry, which essentially feeds the world. The GMO guys feed the world.
Right.
With corn primarily, but there are
soybeans, right?
Yeah, and in fact, they have substantial growing areas here in Hawaii. I remember seeing one in Kauai, but I think there might be others too. A lot of acreage is involved. They don't tell you too much where the acreage is because they're afraid of malicious mischief, because there are those people who, as you know, don't like GMOs at all for whatever their reasons. But the reality is we have GMOs here. There's an enormous amount of capital invested in GMOs around the world. And a lot of people in the world eat GMOs. So if you stop GMOs right now, today, a lot of people would be starving right away.
Yeah, the Hawaii papaya industry would drop dead, basically, because it's all based on a genetically modified variety of papaya that was done to stop this virus.
Yeah, it was a ring spot virus.
Right, exactly.
And Dennis, I'm forgetting his name, but Dennis, local guy, studied at Cornell for a long time. And he made it his business to deal with the ring spot virus and made a GMO papaya. I guess they got by all that legislative attack last year. I'm not sure though. They tried hard, and they were on our show a couple times. And it's sad to see that his discovery that saved papaya as a crop in Hawaii.
Right.
Papaya is more than jobs.
Right.
It's more than a crop. It's a statement of Hawaii.
Right.
It's sweet.
Yes.
You know, and we had a kind of a corner on papaya, and I think we still do, but I'm not sure exactly what the status is. I hope that papaya was accepted from any attack. Anyway, so GMOs can be very useful, and you know, at the end of the day, in order to feed the world, we probably have, at this point, we probably have to have them. But they have a negative connotation, obviously.
They've gotten some bad press for sure from some people.
Yeah.
It's one of those technologies, though, that it can be used for good or it can be used badly. Either way, that's sort of up to the people who control it. But the people who sort of want to ban it at this stage, it's sort of like trying to shut Pandora's box, right? I mean, it's out there already, and it's going to continue. People are going to use it. People know how to do it. You can't stop it now. All you can do is try to appropriately regulate it for the best possible uses in the world.
Yeah, not kill it.
Right.
But killing it would be destructive to the human race.
Right.
But regulate it so that, you know, whatever concerns, the legitimate concerns, not the wild concerns, legitimate concerns there may be in the scientific community are dealt with.
Right. So many years ago, back in the 1970s, you may recall that recombinant DNA came out as this big, it was an emerging technology, and some people were terrified of it, other people wanted to pursue it relentlessly it was going to save mankind. And they finally they actually pulled together a conference at Asilomar, California, and it was a sort of unique conference because it brought together not just a bunch of scientists, but philosophers, legislators, business people, sort of anyone who had something to say about this. And they all sort of sat and talked through all the ramifications, the ups, the downs, and came out with sort of a set of general guidelines on how to pursue recombinant DNA technology in a sensible way.
What is it?
Well, again, it's tightly linked to the same kind of GMO stuff we were talking about.
It's the way you do GMOs.
Yeah, it's the manipulation of
recombinant, you said.
Right. Yeah, it's recombining DNA either internally within an organism or switching in and out genes from other organisms, and and because of that, it has actually been a fairly well-regulated industry, if you will, in the sense that it's produced a lot of interesting, good findings. It's grown. There are nice spin-offs from it, and yet, you know, we haven't had the horrible predictions have not come true. You know,
they have not come true.
Right,
It's
right.
true. You can't find really any
hybrid human-ape kind of things that
monsters,
people,
green slime,
right?
all that.
Yeah, people were talking about.
It's just fiction.
Right, and
so why is there such a hubbub then?
Lots of people don't like change in general. Scientists, I think, while still one of the more trusted groups around, I think are less trusted than they used to be.
Car dealers, politicians, lawyers, and scientists. Right. You join the crowd, Ethan.
But yeah, it is an issue, and the GMO folks have basically they sort of blew their PR a lot. Fundamentally, they've gotten a fairly large angry reaction to GMOs.
But it wasn't always that way. I mean, for example, here in Hawaii, not that you could measure the world by Hawaii, but here in Hawaii, it was like maybe in the maybe 2010, 2008, 2010. All of a sudden, there was some noise, and by 2012, there was more noise. And there's a lot of noise last year and all. But before that, we were growing GMOs, living on GMOs, selling GMOs to the world. Nobody said boo.
Yeah. And
what woke this crowd up? You know,
I actually don't know what stimulated the intensive debate here in Hawaii about it now.
This reminds me of TMT.
In a sense,
you know,
right.
For almost 10 years, the process was going on.
Ground away quietly.
With all the agencies, nobody said boo,
right.
And all of a sudden they get their permit, everybody's wild about it.
Yeah.
I don't understand that.
Yeah. Well, again, science does not exist in a vacuum. Science is part of culture. It springs from a culture and feeds back into the culture. So as the culture changes.
You're right. And the two have to engage.
Absolutely.
That's the lesson of all this.
Right.
Yeah. So, okay. we don't really know why. I mean, maybe this is for another show, why people get so excited about GMOs when there's really no evidence that it does all those horrible things that have crept up in science fiction and in the movies.
Right.
It's the movies. There have been more of these horror movies. And, you think, whoa, green slime, the Night of the Walking Dead, It must be GMOs.
There we go. Are zombies a GMO product?
Okay, all right, now. Okay, so GMOs have a bad rap. They have a rap, and it's probably a bad rap. And people, I mean, they're still anxious about it, and I guess there's a lot of anxiousness going on. Enter RNA. And as I mentioned before with your prompting, RNA is ribonucleic acid as opposed to the old DNA that we talk about all the time in our cells and all that. What is ribonucleic acid?
So again, RNA is a perfectly normal product. We produce lots of RNA in our cells. Every organism on Earth produces lots of RNA. RNA is, in some sense, both simpler but more complex than DNA. There are lots of forms of RNAs. There's transfer RNAs, there's messenger RNAs, there's these things that we're talking about today, the small interfering RNAs. Molecules of various sort of moderate to small size versus DNA, which is a very, very big molecule. It's a very, very long molecule.
Right? What do you call that?
The helix, the double helix, right.
That's RNA with the helix.
That's DNA.
Oh, that's DNA,
Right,
sorry.
in our
and
cells.
the DNA is in the cell.
Right.
You find the DNA with all that information on it in a helix in the cell.
Right. Every cell in our body, except for our red blood cells, basically, has DNA in it. It has probably about a yard long DNA molecule in it.
In every cell?
Each and every cell in your body.
With 48 chromosomes?
I think it's 46, but I don't know.
Okay.
But yes, and remember a few years ago, the Human Genome Project came out, right? And they talked about how many genes, they were going to find out, they're going to map our genes, basically. How many genes do we have? And all the numbers looked really big then. People thought we were going to have 100,000 genes, 120,000 genes. And we came up with a measly, I think, 23,000 genes. People were really surprised that number was that low, because since then they've done lots more genomes of lots more organisms, and
they had more.
Plants have far more than we do. Plants have
the flea had more.
Right, 100,000, 200,000, 300,000 in some cases. I mean, really big numbers, and much more than we do. And
we may have fewer, but ours are smarter.
Well, for one, ours are probably more sophisticatedly regulated, and that's actually sort of where we get into this RNA stuff, because the messenger RNA helps control how DNA gets produced, how DNA produces its products, which are proteins. That's what genes do. Genes produce proteins, basically, but the RNA can tell a gene to produce either more or less of that particular protein.
So the flea may have more genes,
but they may not have as sophisticated regulation of
and
those
the regulation is by RNA.
Right.
The mnemonic would be R for regulate.
You could think of it that way, yes.
And the regulation is what makes the difference. So that's why we can get along on fewer genes,
exactly,
but have smarter genes because the regulation is smarter. And so that lies in the RNA.
Right.
What does RNA look like? Is it also a helix?
Different forms are different shapes, but no, it's not typically a helix. They often show transfer RNA in a sort of a T shape, but it's probably sort of lumped together. The messenger RNA is a very small, 20 little chunks. I mean, they're quite tiny. So
is it in every cell the same way?
Yes, well, all our cells have lots and lots of RNA, and lots of different molecules. I mean, each and every cell in your body is, a monstrously complex factory with hundreds of thousands of kinds of complex molecules in it, churning away, interacting with each other, making new molecules, getting rid of old ones, chewing them up, spitting them out, throwing stuff out, taking stuff in. I mean, every cell in your body is a fantastic
Next time
product.
I clip my nails, I'm going to think of you. Throwing away all this incredibly complex stuff. Okay, so the RNA and the DNA, that is the cells, the chromosomes, the genes, chromosomes that live on the double helix in the DNA, they're all in every cell except red blood cells.
Right.
And now we're going to take a break. When we come back, We're going to talk about this new discovery that appeared in an MIT website only a couple of weeks ago called the Next Great GMO Debate, which is really interesting. And you heard it here on ThinkTech. We'll be right back.
Hi, my name is Cindy Matsuki, and I host the show High Growth with HTDC on ThinkTech Hawaii. This is the show where we talk about all things tech, innovation, entrepreneurship, and manufacturing, because there's so many things going on in Hawaii and more people should know about them. So this is the program that you can come and find out about all the things happening in Hawaii. And this show also airs on Olelo 54 along with ThinkTech Hawaii. And it broadcasts live every other Tuesday at 3 p.m. So don't forget, check out the show Tuesday, 3 p.m. every other week. High Growth with HTDC. Thanks.
Aloha, my name is Josh Green. I'm a senator from the Big Island. I work in the ER there. But on Tuesday afternoons, I get to come and spend 45 minutes to an hour with Jay Fidell and the ThinkTech staff. They're terrific professionals. They help us to bring some of the leading cutting-edge topics here across our state to you. So you can join us at our show on healthcare in Hawaii to talk with leaders from across all the spectrum of health in our state, or you can join us for any other show where we talk about economic development or tourism or some really eclectic programs too. So really, we'd love to see you here on our show. Thanks for joining us and thanks for supporting us.
Okay, we're back. We can hardly wait to get back. It was, we were excited to get back. So this article, I'm going to hold it up just so that you know, this appeared, there's a I guess it's a website called technologyreview.com, and it's put out by MIT, and it's free. And if you want to get on there, you just sign up, and every day you will get great stories about the most high-tech science, technology things imaginable. I get it.
There we go.
Everybody should get it. It's easy. And this came out, I don't have the exact date, but this came out a couple of weeks ago. And it's an article that says the next great GMO debate. Deep inside its labs, Monsanto is learning how to modify crops by spraying them with RNA rather than tinkering with their genes. By a fellow named Antonio Regalato. August 11th is the date of the article. So I sent it immediately to our most likable scientist, Ethan Allen. I said, what? Was machst hier? What's going on?
Well, so the first thing you got to know is, while it is a current article and it's talking about some very interesting recent developments, the phenomena of RNA interference is not new, and it wasn't created by people. RNA interference has gone on for eons. It's part of life. Plants use it routinely and pervasively. It is thought probably it evolved as a way for plants to defend against viruses, because if plants see so-called double-stranded RNA, that's a signature of they're being attacked by a virus, and they had to do something about that, and they figured out what to do, and what to do was to send out these small interfering RNAs, which screw around with the messenger RNA and stop things from being produced. They just turn off genes, basically.
So, this gets really interesting. Who generates the interferons?
Well, the small interfering RNAs are generated by a host in general, yeah.
Okay.
Right.
And then when that happens, a message goes out to the DNA.
Well, no, these actually interfere with, there's a way that the DNA goes through RNA to become proteins, basically.
Okay.
The message gets transcribed or translated, depending on which way you like to think of it. And other RNAs can step in the middle of that process and turn it off by screwing with some of that RNA that's making that transfer. They can also step in and turn the production up if it's a different form of RNA.
Okay, so the RNA, which is sending messages to the DNA, I get this right?
DNA sends messages to the
DNA
RNA, which RNA helps produce the protein.
Okay, so this is, we're talking now about changing the messages.
Well, you can think of it as a volume control. Turn it up, turn it down. Your gene wants to produce at, say, volume level 4. That's what it's programmed to do, and you can get RNAs to crank that all the way up to 10 or to turn it down to 0, depending upon how you like. And you're not screwing with the genes that are producing it. All you're doing is regulating that gene, in a sense.
Okay, so you're not changing the genes. There's no GMO in this process, RNA process.
And once you stop that regulation, presumably after a little bit, the organism will return and crank out the same protein
Whatever
again.
it was
Right.
to begin with.
Right.
So this is the big point. The RNA does not change the genes, the genetic makeup of the organism at all.
Right.
It merely it controls the way the genetic makeup is expressed
Exactly.
by the organism.
Exactly. So it has it's a tremendously powerful tool potentially, right? You can immediately see if you can control aspects of how, for instance, cells behave when they're proliferating wildly in cancer. If you could turn off certain of their vital genes, even briefly, if they're truly vital to their survival, bang, these cells might die. This could be, this technology has tremendous potential to be useful in medicine.
So my genetic makeup controls the way my body conducts itself.
To some extent.
And if I change the way a given gene expresses itself by changing the RNA that in that transaction,
Right.
then I can change the way my body conducts itself.
Right.
But only temporarily.
Right.
Well, not necessarily only, but usually temporarily, I guess.
Right.
And so in the cancer example, as in the virus, the plant virus you mentioned, I can make my body respond differently to an attack by some antigen, including cancer, I guess, by changing the way my genes react to the attack.
Right. Right.
And if I do this right and careful, I could change the way my body deals with cancer.
Right. Exactly.
And effectively, at least in a theoretical sense, I could cure the cancer.
Right. It's an approach that is being examined now in various labs and various places to look and see how this technology can be used against cancer.
Boy, if I was a scientist doing cancer research, I'd be all over this.
Yeah, no, it's a very, very exciting one because it really can get at some of the very fundamental mechanisms. If you can find something about the cancer cells that they are, some gene within them is being expressed that really desperately, those cells need it to be expressed all the time at high levels, and you can turn that off. You've got a tremendous.
I guess they haven't done that yet.
Right, right.
So the idea would be to find a process that is interlinked with cancer, in this case, and then identify the gene that's doing this or not doing it, and then send another kind of message to that gene,
Right.
To turn it up, turn it down, what have you,
Right.
and change the way the body works. But you have to really get in there and find this. Well, there's only 23,000 of them, actually, so maybe it's not that hard.
Well, it's subtle, tricky stuff. I mean, the beauty of it is it can be very nicely targeted like that because these genes are long strings of what we think of as quasi-random letters, 4 different amino acids that make them up. And so if you pick out a string of 20 of those, say, that string is not likely to reoccur in that exact order much of anywhere else. So the beauty of it in the way this article is talking about it and what Monsanto loves is you can target, say, the potato bug, but have an insecticide that will not bother ladybugs or any helpful insects at all. It'll hit potato bugs and potato bugs only, basically.
But you're looking at the genetic makeup of the potato bug,
Right.
not the genetic makeup of the corn.
Right, in this case.
This
right.
is a whole different way to deal with pests.
Right. To make the plant start producing yeah.
Wait. So are you actually changing the cellular conduct of the ladybug or the cellular conduct?
Well,
of
a little bit of
potato?
both. A little bit of both. You're causing a product to be produced that is toxic to the bug, basically.
Okay.
And depending on how you do this, it can either be through something you put on the plant or something you stick into the plant and the plant begins to produce more of it. There's different approaches here.
You go either way, though. You could spray the ladybug, too.
Right.
And you can have the ladybug poison herself.
Fun just to smack it,
Yeah.
You're going to do one by one with the bugs.
The problem is you've got to deliver
Right.
the RNA spray to the ladybug. Maybe easier to deliver the RNA spray to the potato.
Right.
Because you have a field of them.
Right.
So you could have you could get hold of one gene that expresses itself by some kind of poison that would poison a ladybug.
Well, a potato bug, right.
I don't know if poison is the right word, but some kind of chemical that would stop the ladybug, and then the potato would generate more of that.
Right.
And then ladybugs would be stopped.
Right. Part of the issue they're having is with this whole delivery mechanism. How do you get this, either coat the leaves or the part of the plant that's being attacked with this stuff and make it last, because it's a fairly transient compound. The RNA breaks down pretty easily.
Yeah.
Or how do you get the plant to start producing it on its own, so it produces in all of its tissues and or at least in the critical tissues where you need it expressed.
Yeah. That's so interesting. I mean, I remember in the article, which I read only a month ago, that there were really two issues about that. One is that this technique, as far as it exists, is temporary.
Right.
It's ephemeral.
Right.
It's not going to last very long.
Right.
So, I mean, for those of you who would be worried that the RNA is going to have this, multi-generational effect, that's not so. It has an effect for a short time, and then it wears off.
In general, right, when you put it outside, you put it into dirt, and within a matter of a few days, it's basically gone.
No effect on anything.
Well, right. I mean, we, as the Monsanto folks say, we eat RNA all the time, and in large masses.
Part of every cell.
Yeah, right.
But if it's not in the right place at the right time and fresh, I suppose, you want your RNA fresh, then it's not going to have an
It's
effect.
degraded, right? Yeah, degraded.
Yeah.
Pretty fast.
The other thing is, I remember in the article it talked about the good old days where to get a thimble full of RNA that would have some kind of effect would cost a thimbleful, like $50,000, as I recall. But now you can get enough to cover a whole field of potatoes for like $5 or something really cheap.
Right, yeah. So again, we have the technologies now to produce RNA much more quickly, much more in bulk, very specific kinds of RNA molecules. So yes, it's become economically feasible as an approach now to do this.
It's interesting that this would come from Monsanto. You can see them talking at the boardroom.
Oh yeah, yeah.
We got to have a better way than GMOs. Let's think of something, boys.
Right. Yeah.
And they think of RNA, and there's some good scientists there, and they come up with this as an alternative. Why is it appealing? Well, because it's temporary, because it doesn't modify it permanently.
And it can be so selective too. That's one of the real beauty.
Specific characteristics.
Most of these insecticides and pesticides you spread around hurt a lot of animals. They're hurting the helpful animals along with the harmful animals. This stuff has the potential to be very picky about who it hurts, and only will hurt, in theory, the targeted organism.
Yeah. So like the old complaint about pesticides testing affects people and other animals, it doesn't really apply.
Right. I mean, the classic old example was the stuff 1080. I don't know if you remember. The 1080 was a poison for wolves or coyotes. But the problem is then that the vultures that eat the dead wolves or coyotes promptly drop dead from it. The things that eat the vultures promptly drop dead from it. I mean, it's a poison that just keeps on poisoning and just sort of stays there and keeps poisoning for a long, long time and poisons a lot of animals in the environment. This stuff basically, if you've designed it against the potato bug, it should only hit potato bugs and won't hit first cousins of potato bugs, basically,
So it sounds, from what they describe, it's very benign.
In that sense. Now, there's still a lot of unknowns about it, because you are using a gene sequence, and we all have lots of genes and lots of sequences in our DNA. How do you know that sequence that you've picked out that hits the potato bug critically won't, per chance, hit some random gene in, say, the polar bears? I mean, they may have in some part of their DNA that exact same sequence. And we don't really know this. So there are these unknowns about how, well, it looks like it should be very specific.
Well, before we identify all those paranoid possibilities, because a lot of things flow into your mind, let's take a break.
Okay.
And sort of gird our loins for this discussion. And that's probably why, they entitled the article in the MIT Technology Review, "The Next Great GMO Debate." Because there'll be a debate over this too. Why not?
Right.
We're going to take a short break, and then we'll come back and do a little debating.
All right.
And I am the host of Business in Hawaii. Business in Hawaii is a show where we invite guests to come on and explain the secrets of their success for having a successful business in Hawaii. Contrary to popular belief, having a successful business in Hawaii is possible, and there are many examples of that. So please tune in on Thursdays at 2 o'clock, and you can catch us live on livestream.com for our Business in Hawaii show. You can also get the archives on hawaiitechhawaii.com, and we're also on YouTube and also on Oleo and OC16 occasionally. So please join us, find out the secrets of success, how to have a successful business, and I hope to see you on Thursdays.
Okay, we're back, we're live, Ethan and me. Ethan is our likable scientist, and he's a regular host of Likable Science here on Friday afternoon. But because I know so little about this and he knows so much, we made him the guest. And we've talked about GMOs, we've talked about Monsanto's development as reported in the MIT Technology Review a month ago in an article called The Next Great GMO Debate, which is a discussion of RNA, that's ribonucleic
Acid.
RNA. And it communicates with the genes in DNA. And it can get them to change the way they express themselves, change their conduct and therefore the conduct of the organism temporarily.
Right.
And this leads to a whole new way of looking at, may I say, manipulating plants, but maybe every other living thing too.
Right.
And so this part of the show, I mean, it's promising, it's exciting. To me, it's confirmation as we have a long way to go in science. We're discovering the most interesting, exciting things these days. But at the same time, you know, science increasingly has an obligation to look for the community good and
Exactly.
protect the community against danger.
Right. This is why I brought up the Asilomar conference earlier that had been done back in the 70s, the recombinant DNA technology, because to some extent, perhaps some similar kind of event or ongoing dialogue is needed around this kind of emerging technology now.
Okay, well, let's get paranoid. What do you think?
Well, okay, so if this stuff, basically the RNA interference, can be used to silence specific genes, that is production of specific proteins, we have certain proteins, particularly some of our enzymes, that need to be produced all the time to keep us up and living and moving. If somebody fed us the wrong RNA, the wrong interfering RNA, that shut down that production, even transiently, you know, we could drop dead.
In the cells that produce that enzyme, for example.
Right.
Because it could be set up to go right there.
Right.
Just like we talked before about trying to change the way the genes that deal with cancer could express themselves differently and maybe beat cancer.
Right.
But in this case, you talk about the flip side of that.
Right.
Would go to the place, the Isles of Langham, for example. On what is it? What's that?
Well,
enzyme?
I'd probably think of something maybe in your medulla that's having to do with your breathing, because all you
Breathing,
need
that
to
would
do
be
is
interesting,
shut down some key enzyme in that pathway.
Right, so
Once you stop
enzymes make it happen.
Right. Once you
And
shut
if
down
you stop the organ that produces the cells that produce the enzyme, then all of a sudden you're not breathing, and it doesn't take long before you finish.
And then while it's a transient effect, it's had a permanent impact on you as an organism.
And we don't know right now exactly what cells create that enzyme. I mean, maybe we do, but we're a long way from manipulating
And
it
there's
with
a
RNA.
lot of delivery issues still to be.
Yeah.
But
But what it does suggest, though, is a really powerful weapon. You know,
again,
before we have weapons of mass destruction, now we have another kind of weapon of mass inconvenience.
well, it was interesting. I was actually just reading this week's Science. They recently did a rather nice survey of individuals across the nation who have experience in sort of bioweaponry or biological use, biology and the use of defense and warfare. And somewhere close to 2/3 of these experts that were interviewed said that they thought there is likely within the next 10 years to be some sort of a biowarfare or a bioterrorism attack that will kill at least 100 people.
100.
Yeah.
100.
Yeah, so again, it says to me that, again, you're not going to close these kinds of technologies down. The genie's out of the bottle, Too many people know how to do this kind of stuff. If you now sort of try to shut it down so we can't do it, that means the only people doing it are the people who you don't want doing it, or people who are using it for their own selfish or nefarious ends.
It's hard to keep science down these days. I mean, this article,
Right.
there must be, dozens of articles that are right on the frontier that would tell you how to do this. A terrorist could read them like anyone else.
Right. Instead, you need to have these very broad conversations that involve the ethicists, the philosophers, the government leaders, the scientists themselves, the engineers, the man on the street, the philosophers, all talking about really what do we want here? How can and should this technology be used? How shouldn't it be used? How can we best be sure that it gets used only in the good ways and keep the bad uses of it down to a minimum?
But why does the fellow who is at the cutting edge and actually inventing whatever has to be invented, why does he have to listen to any of that talk? It may spur him on to greater efforts.
Well, I mean, there was the guys recently, a year or so ago, who figured out how to, which disease was it that they cranked up I'm losing this. It wasn't smallpox, but it was one of the nastier diseases, and they figured out how to make it air transmissible, which it's never been.
Oh, well, that's so mean.
And they basically had done this sort of as a pure, clean research project.
Because they could.
Yeah, and they actually then held up the publication of that article, and there was quite a bit of debate in the science literature. Should this be published? Should it be published in some redacted form? Should it just be absolutely banned so nobody else?
What do you think?
They ended up publishing it, and they went through a good process and sort of did try to talk this through it and sort of say, how is it realistic to try to shut this off entirely at this stage? And they decided basically no. Again, too much was known. What these guys had done, while very innovative and interesting.
Is there a line, though? Would you say that some articles should not be published absolutely, even though,
That's what this article I was reading on the likelihood of a bioterror attack sort of said. Is there a red line where you say you can't do research in this?
Yeah.
And they're debating that very hotly in what's called dual-use technology now, which are these same kinds of things like this technology, where it can be used for some really good stuff, but yeah, it has potential really ugly effects to it. How do you determine that? You don't want to shut it all down.
I think the world thinks differently now than it did, say, in the Cold War about these kinds of things. There was a big incident in the early 2000s, a protest incident over UARC, something about applied research, a university applied research, and it was funded with the military money, and it was, UH was established, granted, some money to do UARC research. There was a UARC center established here. And the kids came out and occupied McLean's office, big protest on campus. Now it must be 10 years later, and there is a UARC. In fact, in 2 weeks' time, we're going to have the executive director of UARC on our show to discuss what UARC is. I think attitudes have changed about that kind of research, and that the science community has come to believe that it's a global collaboration, and that very few things should be held back.
Yeah, it is, and science has thrived from this globally collaborative attitude it's developed and continues to develop, and the world, I think, is a better place for it. That being said, there are lines of research that you really don't want widely shared because, yes, they may be relatively simple, garage-type technologies that have the potential to be misused rather heavily, and
that could happen here with this RNA.
Yeah, exactly.
I mean, kudos to Monsanto for coming up with an alternative to GMOs, but even a casual reader of this article, the next great GMO debate by Antonio Regalado, August 11th in the MIT Technology Review, even a casual reader would have some concern over the dark side of RNA. I mean, you can stop somebody from breathing. You can stop a field of people from breathing. You can stop a city from breathing. by spraying it, and then it, you haven't changed the genetic makeup of the individual. You just stopped it from conducting itself to breathe.
Right, yeah. I
It
mean,
takes a minute.
Right. But again, it is a long way that the delivery mechanisms for this have not been well worked out. And that's, even the Monsanto guys admit, they said we're still a few breakthroughs away from really making this feasible to use even in relatively benign ways as to do good for agriculture.
Now, just letting imagination fly, if you could change the way our genes express themselves, you could also cure disease.
Right?
Not only cancer, but lots of things. I mean, it's out of Star Trek. Remember they had the table, Dr. That wasn't Dr. Spock.
McCoy.
McCoy, thank you. Dr. McCoy,
Just a
where
simple
are you?
country doctor, as he would always say.
Put him on the table,
Right.
and you scan to find out what's wrong. Sophisticated
Right.
MRI type scanning, right? PET scanning, and then you apply this kind of technology, and it goes to the body itself and causes the body to conduct itself differently, and then presto, conceivably, you're cured of a number of things, and all in a minute. Now, you don't have to wait for the next generation of genes, right. To mutate. You can do it right now.
Yeah, I mean, and they are working on technologies not dissimilar to that, where they will remove some of your blood, treat it appropriately, and put blood back into you that is your own blood, but now is capable of fighting off a disease that would otherwise you would be unable to resist. So I mean, this kind of stuff is going on already, and it's only going to grow and get more and more sophisticated. And the real debate here is how do you regulate it most effectively, so that we get maximum good and minimum harm out of it.
Yeah, but I think, and I'm going further than the article went, I think the real lesson here is not that this is a great substitute for, GMO crops. I mean, maybe they started out thinking that, but it has such great possibilities in so many areas.
Exactly.
Every living being could be affected positively or negatively instantaneously by the way the genes express themselves, that you could really change the world.
Right, and when you think, recall too that when you think of yourself as yourself, realize that you have 10 times as many cells in and on you that aren't your own cells, that are other things living in you and on you, as you have of your own cells. You really are only 1/10 yourself, basically, in terms of the numbers.
Yeah.
So
Sometimes I feel that way too.
if these genes of some of these microbes in you or on you can get changed, that can have impacts on your health and well-being. We're discovering that more and more the health of our closely associated microbes is very important to our own health and how we live, who we become.
The possibilities are incredible.
Right.
I mean, you could change the shape of people. You could make them handsome, tall, athletic. How about that for an athlete?
Yeah.
You could change everything, since the genes control the body itself, if you change the way they express themselves, you could change the whole body.
Having that fine level of control is a very tricky thing, but this is exactly the technology, what they're talking about, is being able, to transiently, let's crank up the growth genes a little bit more here. Oops, now let's back that off. It needs more development of the optical system, the eyes and all. Turn those genes up for a little bit, Yeah, you could, potentially in years, decades, centuries down the road, this type of technology says, yes, you could start doing real designer humans,
Designer humans, that's what it is. It's the boys from Brazil.
Right.
Okay, so that camera over there, that's the people's camera.
Right.
And I'd just like you to address your thoughts to them. What message do you want to leave with them today, Ethan?
Well, again, I think what we've come up with here is that we all need to know about science, and we all should know about science. Science is happening all around us. It's being used. We need to think about it as informed individuals and help make those societal decisions about how it's best used.
Yeah. And for me, I feel that it's happening. I feel my genes are expressing themselves, and they're making me close the show. They're making all the little cells in my body say goodbye. Thank you, Ethan Allen, for a great show.